welcome you all, the last lecture of NPTEL course on immunology. So, I hope all of you enjoyed the course, I mean those who are continuing from very beginning to end, and we tried our best. I mean in this situation, actually what happened, we are habituated to take class in board and chalk system. So, this is kind of not, we are very familiar. Even after that, I hope you understood what I or we would like to tell you, and definitely if there is a scope, we have a live interaction or through forum. You had different questions, and if there is any more questions, I mean definitely we will be here, and our teaching assistant will definitely help you, and we will be here too. So, and in the last part of the lecture or the last lecture, what I am going to talk about, I am continuing the thing what I did in the last lecture. In last lecture, we are talking about monoclonal antibody, right? We are talking about monoclonal antibody. So, now if you have liters of culture of monoclonal antibody, and this is there, I mean there are many other things because they are growing in medium. So, when they are secreting the antibody in medium, it is the medium contains so many other things. So, we have to purify that. Okay. Many ways we can purify that, but the most common technique that we are going to use, and one previous class, I do not remember which lecture it was, but we told that Staphylococcus protein A, the surface protein, okay, Staphylococcus aureus has a surface protein which binds to IgG. Okay, normally monoclonal antibody you go for IgG, IgM monoclonal is also there. That Staphylococcus aureus, that protein A is the surface protein of that bacteria, which binds the constant region of antibody, that mostly the Fc portion of the antibody. So, if this is the antibody, suppose this is the antibody, it will bind the Fc region, okay, it will bind Fc region. So, what scientists did, I mean that is the bacterial defense mechanism, bacteria produce it, but scientists used it in different way. What they did, in a saccharose or agarose bead, they immobilize this protein A, okay, that you can do. Okay, cyanogen bromide activated bead, CNBR activated bead you can buy. So, in that bead, so those beads are actually those beads are activated. Okay. So, they have a free bond, any protein you give it will bind there. So, that way that agarose or saccharose, this is agarose or saccharose. So, agarose or saccharose bead, either one, okay, attached with protein A. So, we will just do the affinity chromatography, very simple. Okay. We have a column, we have just a column affinity chromatography which is packed with that bead. I am not drawing this protein anymore, we have packed with this bead. The same, you know, the gel exclusion chromatography, okay, or gel filtration. So, this is packed with a bead. This bead is a special bead. What? If you see, I mean in amplified or magnified version, you will see that each bead has protein A attached to it. It may not be one, it may be multiple protein A, okay, because beads are bigger or much big enough to hold multiple protein A. So, what will happen now, if you have some solution where you have monoclonal antibody in it, okay, or the antibody in it, definitely the source is one clone. So, it is a monoclonal, you can do it for polyclonal also. Even If you have a serum containing lot of antibody, you can use the same technique, because serum has lot of albumin protein, almost 60% of the serum protein is albumin. So, if you want to separate the antibody from that serum albumin, same technique we use. What will happen? We add this here. Okay. So, we add this liquid here. So, what will happen? It will gravitational force wise it will go down, and if you collect it in a tube, what will happen? So, medium will cross, and while going through this medium, what will happen? All possible antibody are going to attach to this bead, because here the antibody are going to bind. One protein A molecule can bind 2 antibodies. Okay. So, if there are, suppose in this bead there are 100 protein A molecule, 100 protein A molecule in this whole column. So, that means 200 antibody will be bind here, and rest will go to flow through. Okay. So, we do not throw this thing, we save it, because we do not know how much antibody is there, because we can keep it in 4 degree at least for years. Okay. We can keep this antibody solution for years. So, if you pass this, what will happen? 100 protein A molecules are here. So, 200 antibody will be attached here, and rest will go. Then we wash, all non-specific binding or attachment will go away. Then what we do is, so we will take out this and we will put another tube, which is fresh tube. Now, what we are going to do is, we are going to add some buffer, any type of buffer is possible, either very high pH or very low pH. If you just put glycine buffer, normally we put glycine buffer pH 2.5. What will happen? If the pH is too low, suddenly this, because protein A is one protein, it binds the antigen, and this is a protein protein interaction. Okay. One protein is like this, another protein is binding here. Okay. And as soon as you change the pH, either too high like pH 12 or too low like pH 2.5, what will happen? That conformation will change and they will not bind anymore. So, as soon as you add glycine buffer pH 2.5, all antibody, because this is covalently attached, protein A is covalently attached with the agarose bead, but protein A and antibody interaction is non covalent interaction. You change the pH, they will not bind anymore. So, what will happen? When the buffer will come from top to bottom, it will bring antibody also. So, this solution will have all the antibody, clear, but in pH 2.5 antibody cannot stay for longer time. So, what normally we do is, so suppose this is the tube. In this tube, I am telling you very laboratory scale, not the industry scale. Industry scale is completely different. Okay. In laboratory scale, what we do is, in this tube we normally take 200 microliter tris Slale buffer pH 9. Okay. Cl buffer pH is equal to 9. So, if this is here and now something is coming, say 800 microliter, some solution of pH 2.5, when we collect, so total is 1 ml approximate. So, when we collect 800 microliter pH 2.5 and 200 microliter pH 2.5, if you mix it, it will come very much to the normal or the neutral pH 7.2. Okay. So, antibody immediately denature, but we have to renature also, that is it. And now we have multiple tube, you collect them, check them, and you can store it. So, that is how we lab scale we purify. This is the basic principle, like you have to have a affinity purification. Here, where is the affinity? Affinity is between protein A and monoclonal antibody. Okay. So, what I told you? I told you in that bead, activated bead, any protein will bind. So, if you have your antigen enough amount, you instead of your protein A, you can also add your antigen also. That will be much better, because protein A will purify all antibody. Particularly when you are purifying the polyclonal antibody, if you can attach your own antigen of interest, so you will get in the in the collection tube, you will get only antibody which is specific to your protein, no other antibody will be there. Okay. So, that is how we purify the antigen. So, this is one part. So, now you can scale up and produce as much as antibody. You do not have to go to the animal anymore, because you have the cell line, you can store it whenever you need, you culture them, grow them, purify the antibody. So, you do not need any other animal handling or antigen purification, no, you do not need it. Okay. Now, we are going to talk about another thing. It is called, and very briefly I will tell, I will not go detail. This is antibody engineering. Okay. Antibody engineering. So, what is that? We can produce antibody inside the lab just by molecular biology or recombinant DNA technology. What? If you remember, I am drawing again, this is the heavy chain, right. This is light chain attached with disulfide bond. So, which part of the antibody is attaching or interacting with antibody? Only this region. So, what is there? We have one. So, it is heavy chain, variable region, heavy chain, constant region 1 or CH1, this is CH1, this is from heavy chain, and we have another light chain variable region, what we call? We call it VL, and this is VH, okay, and light chain constant region, right, then it is CL. Which part is actually interacting with the antibody antigen? This region. So, now if I make a construct like which will have heavy chain variable region like VH, then there is a linker, then VL. So, if I make only this, what will be there? This is 110 amino acid, you remember your that VDJ recombination class, 110 amino acid, and this is 110 amino acid approximately, it may be 105 or 115, 110 amino acid. Okay, that means how much? That means 330 base pair, here it is 330 base pair, because if you go to DNA level from amino acid, you have to multiply by 3, 1 amino acid 3 codon, and the linker is normally 14 amino acid. This is synthetic. So, what I am going to make is, I am just mimicking the antibody structure. So, I will make one heavy chain domain, there is a linker, and another light chain domain. Okay. So, I will give this 14 amino acid in such a way that this heavy chain binding domain and light chain binding domain are linked with a thread kind of thing. So, they will go and fold. So, we are, instead of making the whole antibody, I am making a DNA construct which will have variable region of heavy chain, variable region of light chain only, which is connected by a 14 to 15 amino acid linker, which is already known. Which kind of combination will fold them better, that is already known. So, we do not have to do research for that. So, ultimately what happened, this is VH, this is VL. So, we are not making the whole antibody, it is a small part. only one part of the Fab, fragment antigen binding, we are making a small part, constant domain also we deleted. So, this is how big will be the total constant. So, this will be 14 means 42 nucleotide, right. So, total will be maximum 700 or very close to 700 base pair construct. So, now this construct is just for a construct which will express a protein, okay. That you can express in bacteria, that you can express in phage, bacteriophage. Normally we express it in bacteriophage, okay. It is also, when we express this, we also call phage display library. Phage display library. So, phage display library can be used for many other things, but it is also used to display the antibody molecule, and this small version of this antibody which has just the VH and VL and synthesized in the laboratory also can do the same purpose. It will also neutralize, cannot do the other effector function, okay. It cannot do opsonization because heavy chain is not there, it cannot do complement activation, but neutralization it can do. It is very small and you do not have to go to animal, you just have to know which VH and which VL, that means which variable domain and which variable domain of heavy chain and which variable domain of light chain actually are going to specific against your antigen. That definitely needs some research, and how I mean time is not allowing me to explain it, because if I would like to explain this phage display library, how to construct from very beginning to end, it will take at least 1.5 to 2 hours minimum. This is not the only thing, because you have to do PCR, lot of strategy are there, then packaging, how what kind of phage, what protein, what to do. So, it will take lot of time, but for this basic class you just have to know that antibody gene can be engineered and we can make it in the lab, because it is, after all, it is a protein. The way we are expressing recombinant protein for all other cases, we can also express the antibody protein in this in the laboratory by recombinant, and this is called, this has a special name, this is called single chain fragment variable. Single chain fragment variable, and in short S small c f v, okay, SCFV. So, the SCFV is this molecule. So, passive, when we need it because more we have the requirement of the passive immunity, more supply of antibody is there required, population is increasing. So, we cannot depend on animal purification. So, new techniques or new things are coming up, okay. This is, and now here is one beauty is there, that when you do the treatment with like antibody raised in other organisms, say mouse antibody, if you use to treat human, it will be treated as foreign, right. So, that need to be changed. So, we have to make a humanized antibody. So, human body should not make. So, if we can replace this Fc portion, making humanized antibody will be much easier, okay. And that is why the SCFB is very popular, okay. SCFB is very popular. So, this technique is very common now and people are using. And one more thing is, I am just going to very quickly I will tell you, is called immunotoxin. I mean I should say this is very important. So, this is one kind of very wonderful strategy after discovering what is there. So, at the end of antibody molecule they put a toxin. Toxin means, suppose diphtheria toxin, okay. diphtheria toxin. So, diphtheria toxin is dangerous, it can kill the cell, okay, it can kill the cell. So, and all this, most of this exotoxin has two part, one is A, it is called AB toxin, which is covalently linked. AB toxin B binds to the cell and A do the toxin part or the killing part. What people are doing and what scientists do, it is actually first proposed and demonstrated by Professor Ira Pastan in NIH, National Institute of Health, USA. So, what he did, his idea was very smart. So, this is the antibody molecule. So, instead of, because B is not doing the doing any harm, AB part, the way I used to remember, B stands for binding and A is the active part. So, A is actually the toxin. So, I used to remember B stands for binding and A is the active. So, what he did, instead of B, so he cut it. So, he attached A part with this antibody. So, he made, this is because this is toxin and this is immunoglobulin. So, he named this whole complex, and which is very popular even today, immunotoxin. What is this purpose? What he did and he demonstrated, now people are trying also for treatment. Suppose this is a cancer cell, okay, this is a cancer cell. So, if somebody has cancer in their body, what is going to happen? Some part is different, cancer cell is going to express some protein which is not expressed by our own protein or own cell. So, cancer cell converted to normal cell converted to cancer cell, and they started producing some antigen which is not present in our all other cell or the normal cells of the body. So, cancer cell will have some extra antigen which is not present, and these antigen is going to be presented by MHC1 definitely. So, what he thought, that if I can make antibody against this which is very specific to cancer cell, now if I inject this antibody into blood, what will happen? That antibody will go and bind to cancer cell only, okay. And now second thought comes, so if this I can make, if I can make antibody which will bind only cancer cell, why can't I attach a toxin here? Okay, why don't you attach a toxin here? So, what will happen? So, the antibody will go and bind to cancer cell which is attached with toxin, and as soon as antibody binds to cancer cell antigen, it will be endocytosed, that means it will be internalized, that is a normal receptor mediated endocytosis that we have talked so many times. So, ultimately what will happen, this toxin will go inside, and as a result cancer cell will die. So, inside the body specifically cancer cell will die, and that was so brilliant idea, and it is, and now it is working. Many patients are now cured by using this technique, and this is very specific. Only problem, you are giving a very bad toxin just in case if it is released, something happen. So, that modality you have to fix. There is a lot of research went on, but in that case you cannot use this antibody from animal source. You have to use antibody from, if it is possible, antibody from that individual person only. So, that will not be treated as foreign, okay. And this is immunotoxin, okay, which was long back in early early 90s, like just around 1890 to 1900, that time Paul Ehrlich proposed the magic bullet. Magic bullet means, he said that we have to find something which will go and kill the target cell or targeted way, so that it will not harm to anybody's any other part of the body. So, targeted, and all of us I mean those who are interested in drug delivery now are thinking for targeted drug delivery. Okay. So, here I will end my lecture here today, and I hope again I am telling you enjoyed, and definitely it was not possible in this lecture the whole immunology. There are in huge amount of interesting thing we could not discuss, because infection immunity is there, immunotherapeutics is there. So, disease wise, so cancer immunity or immunology, HIV immunology, all are interesting, immunomanipulation. Okay. So, many things, but I hope you are now in a position that you can those who are interested who got interest after this course or from before can understand and read these books and the chapters. Thank you all. Okay. So, now, this is the time. So, now this is the time to introduce you all other member who helped us, me and Professor Agnew Ganguly, to make this successful, all this teaching, recording and all other part there, because what you are seeing, it is not only our effort, it is the effort of many other people, and I will request them to come here to just introduce all of you to know that who did the background work. Thank you very much. So, I already told that I would like to introduce the team who worked background and for whom this course has been successfully recorded and you are seeing. So, here myself, you know me and Professor Ganguly. Next to me Shiv Shankar Das, without whom this course would not have been successful, because he is running after me for many years to take a course, and finally it happened. Ramu, who helped me in recording and guiding. So, there is Ramu who helped me recording, guiding what to do, what not to do, how to see it and how to take care of that. Priyanka helped me help us for making question and all other mail and all these things. Saurabh also helped in recording and all other arrangement. So, thank you very much. Thank you all, and many others who is recording right now. You can't see him. So, for all of us, all of them actually, this course has been successful. Thank you very much, and I hope you enjoyed the course. Thank you.